We're coming to the second lecture, this application, this lecture, I'm going to majorly talk about the applications, some of the basic concepts, and some of the scientific breakthroughs, if you will. So a little bit recap from last time. We talk about the keywords of next generation sequencing. So basically, I want you to get the point is there is three keywords. Sequencing can sequence both DNA and RNA. In most cases, so actually we're sequencing the DNA. In some of the platforms, I didn't specifically talk about it. For example, the nanopore sequencer, we can sequence both the DNA and the RNA directly. But for most of the other platforms, you have to convert that into cDNA to do that. And the second keyword is short reads. And depends on the platforms. Normally, we don't do the entire transcripts. Rather, it's for most of popular applications, it's anywhere from 50 base, 100, all the way to 300 base. And in many cases, we can do both two ends. And then the third keyword is obviously ultra high throughput. We can sequence a lot. The second topic I want to refresh your memory of is we had an overview of all the major platforms. Started from the Illumina sequencer, which is absolutely the leading platform so far. And when we talk about the sequencing instrument, especially for second generation sequencing, we need to have two different steps. The first one is amplification. So meaning that one molecule, in many cases, we don't have enough signals. We need to amplify them. And the second one is the sequencing. So for Illumina platform, the amplification is the bridge amplification. You remember I showed you from one tree become a little forest. And then do the sequencing by synthesis. So each step is having one nucleotide comes in with the colorful nucleotide, and then we can take a picture and then get rid of the blockers and then put another nucleotide on, and so on and so forth. So while you are synthesizing the DNA molecule, the sequencing is being measured. So that is a bridge amplification and the sequencing by synthesis. And for the current technology, even for Illumina, they have a little bit change on those, but they just get the point. It's a very similar protocol. The second technology we mentioned a little bit is the solid sequencing, which, well, probably there's only one sequencer now in North America, which fortunately is not us anymore. And the data that being generated, the amplification is in motion PCR. We didn't go any detail about that. And then the sequencing by ligation. I did put a few backup slides for the last lecture. If you're interested, you can take a look at that. And but the major information we want to know for solid sequencing is it doesn't give you the ACGTs as a readout. Rather, it's 0, 1, 2, 3. Well, 0 can be A dinucleotide, CC, GG, or TT. So for every color, it can be represented by 4 possible dinucleotides. So the data interpretation will become difficult. And but I will leave it there. A couple of years ago, I spent a lot of time talking about this because that was still one of the main options for next generation sequencing, not anymore. The third one we talked briefly was the 454 sequencer, which was later purchased by Roche. But this was the first ever commercial available next generation sequencing instrument ever. So it's coming to the market in 2006, so 12 years ago. And I think Illumina was 2007, and Solid was 2008. So that is more like a historical thing. But they do have longer sequencing, and they were based on parasequencing instrument. Another instrument is still very popular. Actually, I will see the sales rep next week. Is from the Pacific Bioscience, and this is very different from Illumina sequencing instrument. Rather, it has a very, very long reads. And these numbers are keep updating. Now it can sequence 30,000 base and probably even longer. And also this is a single molecule sequencing. So what single molecule means is you do not need to do the amplification to do the sequencing. So in the sense that you don't need to make one tree become a forest. But how they can collect enough information is for this technology, they are not say, I need the nucleotide to give me the light to do that. Rather, the polymerase will shine the light once the synthesis happens. So meaning that even if you have one molecule and the polymerase can still have a light, so you can still detect that. There are some advantage of doing using single molecule sequencing, and also that this technology, I forgot whether I mentioned last time or not. This is the only sequencing technology can directly read the methylation signals. So they can treat the methylated C as the fifth nucleotide, and you can directly measure that. So that is a huge advantage for the epigenetic study as well. But similar as some other technologies, especially for third generation sequencing technology, and their error rate are very, very high. So it depends on what we need to use of them. Sometimes if you use that to do the genotyping, identify SNPs, their accuracy is not accurate enough. Another technology is iron torrent or iron proton sequencers, and they're based on the semiconductor-based technology. So basically, once you do the DNA synthesis, every nucleotide you put on, the reaction will release a hydrogen ion. That will change the local pH value. And then we have semiconductor electronic equipment to detect those changes, and therefore you know the sequence. And this was initially marketed as a personal genome sequencer because it's faster than anything else 5 years ago. Not anymore, because everything else becomes faster as well. But it does have a lot of some of the advantage and uniqueness as well. The last one we discussed about was a nanopore sequencer. Remember, this double layer, the inside outside is you cannot. I mean, the electrical signals are different, but you have another protein which is called a nanopore sitting across that double layer, so that when there's a molecule going through that protein, that will change the electronic current that between inside and outside. Because ACGT, they all have different sizes. By measuring how much electrical disturbance that this nucleotide cause, and you will be able to figure out what the sequences are. And of course, this is a very, very portable instrument that has a lot of implication in some part of the world. This is the figure that we will see. Probably you have seen many times from many different studies. So the bottom line is X is the year. So from 2001 to 2017. Y is if you sequence 1 million base of DNA, the cost. Okay. And in 2001, that's the end of the Human Genome Project, it will cost you about $8,000 to sequence 1 million base of DNA. And today, in our lab, it costs about 2 cents to do that. Okay, so some people give an analogy of that is if you fill in your gas tank in 1990, or no, in 2001, and in your car, and now you can drive all the way to the Mars and probably have a round trip of that. That's the amount of energy or the cost that is associated to that. So this figure is, this line is the Moore's law. And the Moore's law is pretty much familiar by the computer science major students. This is something like if every year and the computational cost will decreased by half or things like that. So you can see this is a log scale. Why is log scale? But this is like a straight line. Moore's law is pretty good, right? But you see that this cost initially follows Moore's law pretty well until something happened here, which Illumina introduced their product into the market. There's a huge drop, and there is a few bumps that down the road. So with some additional technology innovation, so the price has been dropped a couple of times again. Okay, so that is the overall structure. So when we talk about this particular lecture, I want to give three major components. The first one is I will go through very briefly about major biological applications using the next generation sequencing. Of course, throughout the whole lecture series that we will talk about the individual technology and their analysis methods. And then I will give you some of the review of the public resources for the next generation sequencing data. So I'm from School of Informatics and my lab doesn't generate any data. Don't worry about that. There are plenty of data you can work on in the public domain. And then I will end the lecture by introducing some of the basic concepts. Well, so the sequencing technology development is pretty incredible. But how we can use that? The major applications including several different components. So the first one is to sequence the DNA. That is not surprising. It is why this technology is being developed. There are several types of applications. So the first one is reference-based resequencing. So what that means is if we are working on humans, or mouse rat, or Drosophila C. elegans. So those model systems, and normally we do not need to, we already know what their DNA sequence are. So our goal is really to see how my genome is different from the reference genome. So those is called the reference-based resequencing. And normally our goal is to see the SNPs, the copy number variations, and the insertion deletions. So our goal here is to see how my genome is different from the reference genome. The second application is de novo sequencing. So meaning that if I'm working on a species that only myself is working on in this planet. So in that case, the chance is I do not know the sequence of that species. So I cannot align my reads back to the reference genome. In that case, the analysis will be completely different. That is called de novo sequencing. And we will have some lectures talking about the methodology to do those analyses. So the third one is called metagenomics, which is not sequencing the human DNA anymore. It's coming from a piece of either soil or seawater or our skin or the gut or whatever, and then to sequence to identify who is there in the mixture of microbiome species. So basically there's many different types of bacteria in those, and we want to identify who are in there. So those are called metagenomics, which I will give you a little bit more introduction in the later slides as well. So the second type of application is to sequence the RNA molecules. Because we can sequence so much and so fast. So one of the major applications will be the RNA sequencing technology. We can use the same technology to do the novel non-coding RNA identification as well as the small RNA identification. The third type of technology is to study the protein DNA and the protein RNA interactions. And which the technology is called either ChIP-seq, if you work on the DNA binding proteins, like transcription factors, RNA polymerase, or histone markers, or CLIP-seq, if you are working on the RNA binding proteins. And by combining everything together, we can always study the epigenetics as well, including the DNA methylation, histone modification, nucleosome positioning, or chromosome looping. Okay, so this is the chromosome looping architectures. And this year, for the first time, we'll have one lecture fully devoted to the chromosome looping type of research as well. It becomes very, very interesting. Okay, so now let me go over, give a little bit more detail about the individual type of applications. So the first one is the DNA sequencing. For the people in our medical school, the majority goal is to do the resequencing, the reference-based resequencing, which our goal here is to identify the genetic variants comparing to the reference genome. And there are a couple of different types of signals. The first type is going to be the single nucleotide variants, or SNV. So over 82% of our genetic variants are the SNPs or SNVs. There's a differences in that terminologies we will talk about when we get to that part of the lecture. But the general idea is pretty simple. So this is one nucleotide reference, and my genome is different. That is a single nucleotide variance. So the second type is this, it's called small insertion deletions. And you can see from the figure, this is an insertion case. The top line here is the reference genome. And in my DNA, there is an additional 3 letters are being plugged in. So this is called insertion. In some of the cases, it's called deletion. And this is something that represents almost the other 18% of genetic variants. And actually, this small index will give us some of the problems to do the identification. And we will go through those in the later lectures. The third type is the large structure variations that will include the copy number variations, large insertion deletions, inversions, and gene fusion, and so on. So we can give a little more detail in the future lectures. So that is the type of variants we want to identify. In terms of the DNA sequencing, the scope, the first scope is going to be sequence the whole genomes. And so we know that in our DNA, we have 3 billion letters. And this picture shows in 2008, this is published in Nature. So you can see this gentleman here is James Watson, right? He found out the double helix. And so it's, I think it's just very much justifiable. The first individual human genome is the sequence of DNA of the James Watson. And at that time, the work was done in 2007. They used the 454 technology, did the sequencing. It cost them $2 million to sequence the whole genome. And at that point, they have 7.4-fold coverage. So what that means is that we will get into much detail in the later lectures. So if we want to call a SNP of an individual DNA, it's not just because we identify, we say, oh, in this piece of DNA, there's one nucleotide different from the reference, we'll call it. No, because this technology is very accurate, but it still have 0.1% of chance to make mistake. I mean, that is a lot of mistake if we're talking about 3 billion letters. So what we need to do is we will have multiple measurements for each piece of the DNA, and then using that to figure out the DNA, the SNPs. So far that our standard is we will do about 30x coverage. So for every nucleotide, we will at least have 30 sequences to cover that region so that we can have a confidence call. But now, so you see, at that point, they did a 7.4-fold coverage. And they were still able to identify over 3 million SNPs between the Watson genome versus the reference genome. And almost 11,000 of those are non-synonymous SNPs, meaning those are the SNPs that will change the protein sequences. Okay? So, I mean, we know that one of those make him smarter than the rest of us, but the issue is we don't know which one was that. But anyway, so you can see that this is the first scale, is to do the whole genome sequencing. Of course, we are never satisfied. And then shortly after that project, the 1,000 Genomes project was launched. and which by name you can see is sequenced 1,000 genomes. And the goal here is really trying to identify the deep catalog of human genetic variations. Okay, so what that means is in the Human Genome Project, which was the 1990s project, by the end of that project, we know how to define a human in terms of the DNA sequence, but we don't know how variable our DNA are. And this project is trying to see that between different populations, within different populations, how variable our DNA sequence are. And eventually we sequenced actually 2,500 individuals, which is more than 1,000, and which was 500 genomes from each individual, each of the 5 major populations. So we'll talk about that a little bit as well. And also there is a Cancer Genome Project, which is TCGA, or Cancer Genome Atlas, or ICGC, International Cancer Genome Consortium. But the point is the first level of the scope of the DNA sequencing is whole genome sequencing. Well, whole genome sequencing is great, except for it's still very, very expensive. But sometimes when we have very limited funding, and what we're going to do is we cannot focus on the whole genome. Rather, we want to focus on a piece of the genome. So there comes to many different strategies. So the first one is to do the whole exome sequencing, because if I know nothing about this disease, I better focus on the protein coding region part of it. So that's the justification, which they go for the whole exome sequencing. And sometimes you can also focus on the region that is more being captured by linkage or GWAS study that was published earlier. So you see, oh, for example, for this particular case, from our earlier study, we know that there are something in this region associated with the disease. Let me sequence a little deeper in these regions. So identify a region of genome that you want to sequence. Okay, so that is called targeted sequencing. But it sounds simple, okay? How do we do that? We got the DNA, and we cannot just pick out what are the exon part of it, and we need to have some technology to do that. The way to do that is there are several different technologies that will include a long-range PCR, and this is only for a small part of the capture. You're only interested in certain regions. You can do that. And there is also reduced representation library. So historically, people have done that. But the real thing that people are using now is a so-called capture array or capture in solution technology. Okay. So we will talk about that later regarding the technology. How do we really do that? Basically, you need to design the probes to target on those regions, and then put them down, and then to do the sequencing. Cancer genome is another scope that only focuses on the somatic tissues, right? A lot of people say cancer is a disease of the genome, and in many cases, it's have a higher rate of abnormalities, and sometimes more than 10,000 mutations per cancer. And very few of them are the driver mutations, and the rest are passenger mutations. One of the goals is whether it's possible to identify those drivers in the tumor that drive the cancer progression, right? So this is a cartoon that this is the little angel that God, the human genome code has been unraveled. And this apparently is the God said, damn those hackers, now I have to change the password. So this is actually exactly what happens in the cancer genome scenario. And the real thing we want to identify are those drivers. But the problems are so many passengers. They are there. They are the password from the God. So we don't know who is driver, who is passenger if you have so many passengers. So one of the things that we really need to understand are how to select the driver mutations so that we know better about the disease and the cancer progression and potentially design the drugs as well. Okay, and sometimes there are therapeutics based on the genomics. So here is one example, and very early. So the example I gave is very early. It's 2010. So you can see that in this particular example, is they got the patient got a tumor, and then they did genome sequencing, and they were able to identify the structure variations for this particular cancer type. So you can see there's those are blue rays, those are coming from the blue chromosome, and red are coming from the red chromosome. But red chromosome and blue chromosome, they don't supposed to be together. They should be two separate chromosomes. But because of this cancer, there's a chromosome rearrangement. They did identify some of the rays are like that. So one end is in the blue, the other one is in the red. And then once they identify those, this is going to be their biomarker. Okay, so what they do is they will do different treatment on the cancer cells, and eventually they use this part to see how much cells will I still observe this type of chromosome rearrangement. So after a few treatment, so you can see this signal are going down. So this is one of the strategy to use this technology, identify this patient-specific biomarkers so that you can trace back with the therapeutics, and potentially you can observe the relapse or I mean having those monitoring those events as well. Okay, so that is the DNA sequencing part. RNA sequencing is much more straightforward. You got the RNA molecule, and this is a messenger RNA. It has a polyadenylation on it, and so what you do is you break the RNA into smaller pieces, and you put the Illumina sequencing adapters, and then go ahead to do the sequencing. After sequencing, so you can see some of the reads are still have a lot of A's there, and those are potentially the one that close to the end. And then what we do is we will map those sequencing nucleotide into the genomic DNA. So some of the reads are mapped to the exonic region of it. So you can see this is exon, this is the intron. Some of the reads are like this. One end in this exon, the other end in this exon. So these are the ones that we call the junction reads. It's super useful for us to do the splicing analysis. And then for this part, towards the end, you can see there are poly A reads as well. So we need to just, after that, you will understand the biology a little bit better. So what we get out of the RNA sequencing? I mean, of course, the first goal to do RNA sequencing is to measure the gene expression. So you can see that in this case, this gene expressed this much, they are expressed this much, and so that is something we can measure using the RNA sequencing. But the gene expression difference is not the only thing that is variable on the transcriptome level. There are several other things as well. So the first one is the so-called splicing variance. So you can see that we got a gene, this hypothetical gene has 3 different exons. In one condition, all 3 exons are in the final gene product. In the other condition, only the middle one is gone. Okay, so these 2 products, they may have the same level of transcription level, but because of this splicing changes, you can imagine that the protein function will be different as well. So this is another dimension of the variability of the RNA molecule, so that people will use this technology. They will have a tool to identify those. The third level is, in addition to the expression level and splicing variance, because we are sequencing the RNA molecule, so it's not only counting how many reads are there. Now we also know what are the sequences of those RNA molecules. So you can see in this case, in the reference genome is G, and the RNA being transcribed are half of G or half of A, and you know this is a heterozygous variance in this particular location. If you do the analysis properly and with the data properly, you can study allele-specific expression, you can study RNA editing, and you can study a lot of things you would not be able to dream of using other technologies. So just need to be creative and exhaust what is being buried in the data. So what we can get out of the RNA sequencing experiment? You can do the gene expression, we know that, and then you can look at identify differential expression. So when we go to the RNA sequencing lecture series, this is one thing that we certainly will look at, okay? And there are a lot of smart people figure out the statistical models that are ready to be used to do this type of analysis. As I mentioned earlier, the second thing that we can study alternative splicing. And we can also look at the variance on the transcriptome levels. So you can see in this case, this is going to be the heterozygous variance. And in another case, this is the heterozygous. So the previous one is the homozygous variance. And this is the heterozygous variance. So once we identify those variants, we can do a lot of other things as well. We can use a different level of information technologies to see which are those variants will change the amino acid sequences. They are called non-synonymous SNPs. So once it's being changed, whether it's going to change the protein function or protein structural functions. So we will go through all those tools that we need in this course about how to do those type of predictions. The second level is if you are talking about a SNP that is pretty silent. So basically, it doesn't change the protein sequence. In many cases, people will think, oh, they are synonymous SNPs. They are not useful at all. But actually, they may impact the RNA binding protein binding and change the splicing regulation. So those are the things that we will go through as well. So the third level is if some of these SNPs locate in the 3' UTR regions of the gene, they can readily interfere with the microRNA targeting and further coming back to impact the gene expression through the RNA degradation. And if we are really creative, if we are really hungry, and then you can bring this RNA binding protein information with the splicing or integrating the microRNA sequencing, the microRNA binding site data with the differential expression analysis. And that will be very useful as well. And as I mentioned earlier, that properly use this sequencing information, you can study allele-specific expression as well. And we have some paper just on being, we were finalizing the paper, and it will give you very, very powerful analysis, which we will go through some of those analyses when we get to the RNA sequencing side. And the last one is you can also use this to study non-coding RNA. This is something that microarray technology will not be able to, because for a lot of non-coding RNAs, we don't have their annotation. We don't know they were existing there. So nobody designed microarray to target on those regions. Well, with RNA sequencing, whether you know they are there or not, as long as they're there, you'll be able to measure it. You can always coming back to understand your biology a little better. So there's a lot of stuff we can get out of the RNA sequencing. Next part, ChIP-seq. ChIP-seq also stands for chromatin immunoprecipitation following next generation sequencing. Okay, so the goal of ChIP-seq is to identify genome-wide binding patterns of a protein of interest. Which means that if I have a protein that is either a transcription factor or a histone marker or RNA polymerase, I want to know on the DNA level where this protein binds. Okay, so the way we do that is we got this, you see, this is initially what happens in the cell. You have this protein you're interested in, and then double helix binding there. So what we do here is you do two things. One is you do the cross-linking. So the cross-linking meaning you normally use formaldehyde, or you can use UV light as well. So meaning that when the protein is binding on the DNA, and after this cross-linking, they cannot be separated anymore. So it's like you dump a lot of glue in that, so it's fixed there. And then the second part you do is you fragment the DNA or share the DNA into smaller pieces. So you can see some of the DNA molecule have the protein binds on that, others are empty or binds by other proteins. So next step, you are using an antibody to target on this protein of interest. That's why you have to have an antibody to study this. And then what you do is you wash everything else away. So meaning that only the DNA molecule that initially bound by this protein will be left in this environment. And then you don't need the protein anymore. And then you got the sequencing, and then with the proper treatment, we can do the sequencing analysis. By the end of the day, by doing the analysis, you'll be able to identify where this protein binds in the genome-wide scale. Similar assay exists also for the RNA protein interaction as well. ATAC-seq is another technology. So this is the first year we will devote some of the lecture to talk about that as well. So it stands for Assay for Transposase Accessible Chromatin Using Sequencing. Those never make sense. But you see what this assay does is we know that the DNA are being wrapped around by histones, and there's a part of the DNA that are closed, and then there's a part that are open, right? And if we want to start a transcriptional initiation, and that part of chromatin needs to be opened. Otherwise, transcription factor cannot get in, RNA polymerase cannot get in, the transcription is not going to be started. So identify which part of the chromatin is opened or closed. That is super, super important. The way that this assay do is they will use an enzyme called TN5, which is called TN5 transposon. So what they do is, this is kind of the enzyme that virus is using that a lot, and that is how the virus put their DNA into our host DNA, right? But we take the advantage of this enzyme by engineer this Illumina adapter into it. So what you do is, if you treated your DNA or chromatin with this enzyme, modified enzyme, and by the end of the day, you will insert Illumina adapters into your DNA, okay? And then we can PCR them out and sequence that part, right? So through this way, the trick for this is if your chromatin in certain part of the chromatin is opened, if the transcription factor can get in, or if RNA polymerase can get in, the chance is this TN5 can get in as well. So they will go there to insert the Illumina adapter into that, and then you PCR them out, and then you do the sequencing. For the part that is closed like this, and the TN5 cannot access to the DNA as well, so they cannot have any function. By the end of the day, we don't have any signal out of those, okay? So this is a comparison of different technology, but the point is using this technology, you can start from a very small number of cells, and sometimes about 500 to 50,000 cells will be adequate. And this is what is being showed in this. This is ATAC-seq, and this is using DNase, and this is kind of using ChIP-seq. So you can see using ChIP-seq, targeted on CTCF protein, you are observing a big peak here. That means CTCF is binding there. That means that part of chromatin has to be opened. So what you can see is in this region, there's a lot of signals being identified, right? And then, but if you look at this in a little bit more deeper thinking, give a deeper thinking, say, what happens here? Why this part is not being there's no signal? Because CTCF is binding there, so TN5 cannot get in as well. So it has a protection effect as well. So by this, if you do the analysis properly, for some of the transcription factors, you can identify their target, their binding sites as well. So this is some of the data from our lab, so you can see after you do the assay properly. So this is using the ATAC-seq. So you can see all those genes, and here is the promoter regions of the genes. So you can see this part, we see a very, very strong signal normally. That means that part of the chromatin is being opened. For some other genes, when they are being closed, and you wouldn't able to identify those signals, okay? And you can also compare whether this chromatin open and closed between different conditions, whether their level is different. Right? So you can see this one is It's open, but not that much. But after treatment, this part of chromatin is completely open. So you can see a lot of signals in those regions as well. And I think I showed this last time, did I? No? You did? I did? Okay. I gave a lot of lectures. Sometimes I forgot in which course that I showed this or in which course I did it. So this is a little bit zoom in of the signals. As you can see, there's a big hole. And this hole is because of there is a transcription factor binds there, so that left this footprints here. Okay? And this is based on the frozen samples. So I'm not going to repeat this. Okay, so those are a very high level of overview of different things that we can do using this technology. Of course, we have not touched base on the analysis at all, right? So we will start those from in the next lecture, which is on Monday. So the next part of this lecture, I want to go over some of the public resources for next generation sequencing data. And most of them are based on large consortium work. And through those, we not only have the access to the different type of NGS data, more importantly, there's a lot of science being figured out behind those consortiums as well. So the first one is the Southern Genomes Project. And I mentioned this earlier. The goal here is to provide a deep characterization of human genome sequence variations. Through Human Genome Project, we know what are the sequence define a human, but the goal is to see how different we are. And eventually they sequence 2,500 individuals and 500 each of the major ethnic groups. So you can see they are listed here. And the data is online. The VCF file is there. We'll talk about what the VCF is. But the variant signal is here. The original files are there as well. And through this project, there's a lot of data being generated. There are many tools are being developed, like the GATK package that is our standard to do the DNA sequencing analysis. And many standards are being generated in that consortium as well. So there's a huge impact for the scientific community. And this is a few articles. So basically, they were able to identify many SNPs, many small insertion deletions, and many structure variations. And they also found that each person carries 250 to 300 loss of function variants in annotated genes, and 50 to 100 variants implicated in inherited disorders. Well, you can read all these findings left and right. But the point is, Southern Genomes data, their patients, these 2,500 individuals, they're healthy individuals. They don't have apparent disease, at least at the time that they were being sequenced. Which means, if you read through this, it's kind of surprising. Like for these healthy people, there are so many variants that potentially causing disease are occurring in our genome. So all this really tells us that, yes, we can have the technology to sequence our DNA. We probably don't know how to properly interpret the result yet. And this is part of the informatics work that we all need to figure out, and including the wet lab experiment and to understand the mechanisms. The second project I want to talk a little bit about is a GTAX project, or Genotype Tissue Expression Project. So the goal of this project is to provide a reference data set to study genetic changes in the gene expression. So basically, we know our DNA, unless in cancer situations, all the cells in our body, the DNA should be identical, right? So DNA is like our blueprint that if we sequence, we got it. But RNA level is different. And because for different tissue, different gene will express. But how do we know, let's say if we study breast cancer, how do we know what should be the normal gene expression in the breast tissue? and for liver, for kidney, and for everything else. So in this project, they were really trying to see that in the normal condition, what are the gene expression levels that in our different tissues. So this project was launched in 2010, and so last week I was preparing for this lecture, so I checked this. They eventually identified over 700 donors, and those are postmortem people. Most of the pass away by accident, and some of the is a cardiac events. And what they did is they got the person they know they are passed away, and they have to rush into the family to have the consent, and then to donate the body into scientific research. And they were trying to get many tissues, as many as possible from these donors, and then they will run the RNA sequencing and a few other assays on that as well. So it's incredible. I mean, it's really incredible. So they got over 700 donors across 50-ish tissues, and they were eventually get over 11,000 samples, and they did RNA sequencing. So you can see that for different tissue type, this is the number of samples they have. This is provide an incredible resource for us to understand the gene expression levels in different tissues. And the other demographic data is shown in this slide as well. Well, so for these donors, they not only identify their tissues and then to do the RNA sequencing, they also genotype them. I think for some of them, they also did the other level, like DNA methylation studies as well. And one of the things that they want to find out is the EQTL analysis. Basically, EQTL stands for expression quantitative trait loci. So we will Dongbin Lai, which will teach the statistical genetic lecture, and I asked him to give a little bit more in-depth definition or introduction of the EQTL analysis. But the general understanding for that is, so you can see for this individual, for this gene, and in the promoter region, these three individuals, they have different genotypes, right? This is AA, AG, or GG, okay? And so these are from the DNA level, they know their DNA sequence are. And then if for different tissues, and then they have the measurements for the gene expression levels, and they can do the association between this genotype with the gene expression levels of different tissues as well. So they will see whether this is associated with the expression levels in blood or in liver or in lung or both, all of them. So if there is, that means this gene is undergoing cis-actin regulation. So this is something called cis-EQTLs. And there are some of the scenarios called trans-EQTL as well. So those are the information they were able to figure out from the GTAX project. And because they have the gene expression levels of so many individuals on different tissue types. Okay, so the publication, there is a Nature collection of publications last year in November, and I encourage you to take a look at. And the processed data can be found in the GTAX data portal, and which in many cases directly download format. You can download the gene expression levels, they normalized. And if you do need the raw data, you probably need to go through the dbGaP database, which I will introduce a little bit in the later slides. So that is the GTAX. Okay, so the 1,000 genomes give you the DNA level of reference. The GTAX give you the RNA level reference. Those are all great. We know our sequence now. But how do we know which part of the DNA represent what functionalities? So now comes to the ENCODE project, which is the primary goal is to annotate our genome. Now you have all the sequences, but how do I know this is the paragraph that I need to read? So you can see that from this chromosome, and then if you look at it in different ways, in different scales, this will be wrapped around the histones, and then this is open chromatin regions. In these regions, there is a histones, so you can see the wrapping in that way. And some of the DNA is going to be methylated, and there is an RNA polymerase is working here, which means there is a gene here. And then after being expressed, it will be transcribed into the RNA molecule, and there's an RNA binding proteins. So the GTAX, the ENCODE project is trying to figure out the different level of regulation, how to read this DNA book. It started from 2003, initially using primarily on the microarray data analysis. In the very first phase, they identified 1% of human genome they try to understand. And later, because of technology development, they will just focus on the entire genome. So these are the technologies they have been taken, and actually it's an incomplete list. So that will include 5C ChIA-PAT, high-C, those are for the chromosome conformation, which we'll talk about in the later slides. DNase-seq, FAIR-seq, and ATAC-seq. ATAC-seq will be the major thing now, but in the past it was DNase and FAIR-seq. ChIP-seq to study individual histone markers and the transcription factors. And whole genome bisulfite sequencing, reduce the representation bisulfite sequencing, RBS, or methylation arrays, understand the methylation signals. And the RNA sequencing identify which part of chromatin is being opened and transcribed. And then CLIP-seq or RIP-seq is trying to identify the RNA binding proteins. Of course, there is a lot of efforts on the computational annotation as well. So now I think they also add in something like RNA methylation. There's a lot of those level of analysis as well, but it's super, super useful. And the data publication collection is published in 2012, and I think soon they will have another collection because of the new phase of the project. And the data can be accessed through this website, and the tracks, all the tracks of those are available through the UCSC Genome Browser and other browser as well. Okay, which Guanglong will talk about in the next couple of lectures. Similarly, ENCODE is primarily for human, and there is a small percentage is for mouse. But there is a couple of groups of people and trying to do the mode in the ENCODE, which is focusing on Drosophila and C. elegans. So it's kind of in parallel with ENCODE, and there's a lot of understanding of the genome annotation of those two model organisms as well. And another similar project is Epigenomics Roadmap project. So the focus is very similar to ENCODE in terms of overall scope. It's trying to annotate the genome. But the focus here is it's focused on the stem cell and the ex vivo tissues. Okay, ENCODE, I forgot to mention, most of them are using the cell lines. Okay, that's what the ENCODE is more focusing on. For this one, is more like an in vivo type of or ex vivo type of tissues. So they measure the DNA methylation, histone modification, chromatin accessibility, and small RNA transcripts. So you can see that when I make this lecture a couple of days ago, I put I just checked their website, so you can see those are different tissue types. And again, these are not cell lines anymore. Those are tissues. And here are all different type of histone markers. And so you can see the matrix, see how many data they have. Again, all of data are available in the public domain that you can download and do the additional analysis. The next one is the TCGA project, or Cancer Genome Atlas project. And this is a more cancer-oriented project by name. You will figure it out. And it's a huge amount of data. So the goal of this project is to understand the tumors. Okay, so taking breast cancer as one example. Now, we can do the DNA sequencing, but DNA doesn't tell us all the stories. We want to know as much as possible about this particular tissue. So what they did is they not only look at the DNAs, including the germline and the somatic mutations, but also the RNA signals. They have done initially microarray and then later RNA sequencing. And they also did a microRNA sequencing. And there is a DNA methylation signals. There is a proteomics technology being used as well. So the goal here is really for major cancer types, they want to understand the molecular features of these major cancer types in a much deeper scale. So you can see they have done a lot, and there's including 33 tumor types, 11,000 patients, and again, all the data is in the public domain. And the data access, initially they have a data portal, but later becomes the Genomic Data Commons, GDC data portals. So here is where the actions is. And also that if you need to access for the processed data and the raw data, this GDC data portal will be the place to go. And they actually break the data into different levels. They have tier 1, 2, 3, and 4. For tier 3 and 4, those are much processed data, and you can directly download, okay? And for tier 1 and 2, you need to have certain permission to do that because the DNA sequencing has the patient genomic DNA in that, although it's tumors. So you still have that there's a lot of privacy issues that are involved, although it's de-identified, but still there is a way that to trace back to some of the history. So that's one of the reasons you need to have certain permission to download data. It's achievable, and it has been done very broadly as well. Another project that is very relevant is called ICGC project, International Cancer Genome Consortium. And again, by name, you know this is a cancer project. This is the international effort. And now TCGA become one of the components in the ICGC. So you see in the United States, there are other cancer types being focused on in Canada and Europe, different countries, and in China, Japan, Australia, and in India. So they all different countries are focused on different type of cancer, more predominant in their populations. And the data I have not tried, but I think in many cases it's downloadable as well. Okay, so those are cancer. Okay. So now another database that it sounds interesting is called the Genome AD or Exact project. This is sort of new. So the Genome AD is called the Genome Aggregation Database. And Exact is actually the first phase of the Genome AD database. So basically, what they are trying to do is we have the 1,000 Genomes project, and it's so annoying to have many different groups. They have done so many whole exome sequencing by now. And if I want to look at the data jointly, I need to download every single one of it. And then I need to reprocess the data in certain uniform way so that there's as little batch effect as possible to make the data more comparable. So it's almost become impossible to do, right? So what they are trying to do is that they were trying to put all the sequencing data being generated and available here. So the current data set by this month has 123,000 exons in that, and also 15,000 whole genome sequencing. And these are from unrelated individuals. And the sequence, their data coming from different consortiums, including 1,000 Genomes, ADSP, GTEx, and TCGA. Those are the few things that I know about. And actually, they have a long list of the consortium that contribute their data into the Genome AD project. By the end of the day, it becomes extremely useful because all the raw data from different projects has been reprocessed through the same pipeline and jointly variant calling that being offered. So this can offer a very, very important resource for us to do a lot of studies. The next project is the Cancer Cell Line Encyclopedia project or CCLE project. And so they provide the public access to genomic data analysis and visualization about 1,000 cancer cell lines. So by name, you know they are focused on the cancer cell lines. And they identify a couple of different levels of information, including the DNA level, more on the DNA copy number variation and the SNP array data. And this is important. They also did a lot of RNA level analysis, and I think most of them have the RNA sequencing data available, and also some of the mutation data. And so the important part of this is it's not only the RNA sequencing of 1,000 different cell lines. Through a different project or CTRP project, and they also did the drug-resistant, drug sensitivity response data from over almost 500 drugs or compounds. So you can think of, we have 1,000 RNA sequencing put aside, that's from CCLE. But in the CTRP, for this 800 cell lines, and we have 500 drugs, so there's a lot of combinations in that, and we know their drug response data as well. So that will offer incredible resource for us to understand how the gene expression level at the basal level can help us to predict the drug sensitivity, whether it's possible or not. So those are the data that is being available in the public domain as well. Another related project regarding drug is the NIH LINKS program, and this is called the Library of Integrated Network-Based Cellular Signatures. And so this is slightly different from the CCLE. The CCLE is the expression level is in the basal level. Okay, get a cell line. I mean, I measure the gene expression, but if I treat with certain drug, I do not measure the change of the RNA level because it's too many combinations. For the links, they were trying to solve this problem. So they have a lot of combinations as well. So you have so many cell lines, you have so many drugs. So for each drug, each cell line, you treat before and after, you measure the global gene expression levels. That's a lot of data. That's why they cannot do it. And they eventually, the way they did that is they call it a L1000 platform. So basically, they have some way only measure 1,000 gene expression levels in this before and after drug treatment. And then they also have a way to try to impute other gene expression level based on the ones that are being measured. So there's a huge resource in the public domain. And in 2017, when I checked the Cell paper, they already have 1.3 million publicly accessible gene expression data available in that consortium. So if that's where your research will lead you to, that is an incredible resource. Another database that I think most of you already know is the GEO database. This is where people put their gene expression data into, and including the microarray and the RNA sequencing and other types as well. So they have array and sequencing based, but they also provide you to have a Miami format compliant data submission, which means you are not just dumping your data into the database. You have to tell them everything about your data set, including what are the tissue type, how you get the tissues, how you did the RNA extraction, what are the protocols, if you did RNA sequencing, what are the kits did you use, and then how did you do the alignment, how did you do the gene expression quantification, and so on and so forth. So it's a very, very intensive metadata are associated with every single submission. And they have their staff, they bug you. I mean, if you don't really provide this information, missing some information, you cannot submit into that. But you may say, why I have to submit? Well, this is required by many journals. If you don't submit, and your paper is not being published. So and this is something that, I think that is a really good thing that for the scientific community, so that we can have a larger access to many data sets. This is super important. There is another similar resource in Europe. It's called the EBI Expression Atlas. I haven't never tried to get into detail of this, but it's also another very credible source as well. Another database is called dbGaP, and this is the database of genotype and phenotypes, and this is a more protected data sets. For the GEO database, you can deposit RNA level data into that. Normally, it's okay. But if you want to put the raw data in, if it's a cell line or animal study, it's no problem. If it's a human study, and you might have some legal issues because in theory, you can identify the genetic variants from the RNA sequencing data and trace back to the individuals. So that's why that even in the TCGA project, in the Cancer Genome Atlas project, for the RNA level data, if you want the raw data, you have to go to the protected data sets. Okay, and the dbGaP is one of those that has all the DNA level and RNA level sequencing, original sequencing data in there. For example, dbGaP is in the GTAX project is in the dbGaP here. So they have a lot of molecular data and then phenotypic data as well. So you, as a graduate student, you cannot access the dbGaP database. It has to be initiated by the PI, and then it has to be signed by the university officials. And you have to provide a proposal to access the different data sets. And so basically, they will require someone can fire the PI to sign that. So in case this data is being released to someone that shouldn't have the access, that person will get into trouble. So there's a lot of protection going on. So if you want to access anything inside the dbGaP, you want to work with that out with your PI. Okay. The last public resource, there's a lot of public resource, right? This is an incomplete list, actually. Is the NIH Human Microbiome Project. And this is the one that the goal is, by name, that it's trying to identify the microbiome species in the human body. And did I talk about that last time? About micro? No, okay. My brains are kind of scattered in which course I talk about what. Okay, so microbiome is incredibly important, and it's the size. So you see, I'm standing here, right? You are assuming that everything you see is human cells. Actually, there are 10 times more other species cells than human cells in our body. So I think it's written here. Bacteria in the average human body number 10 times more than human cells. I mean, their sizes are small, they're tiny, and they're accounting for about 1 to 3% of the body mass. And the overall weight is very close to the brain. And so a lot of people consider that microbiome is another organ of a human that we have never systematically studied. And in terms of genetic variability, they offer 1,000 times more genes than human genes because they have so many species, variety in our body. And so they are everywhere on us, and on the skin, the nose, in the GI tract, and everywhere. So in many cases, they are helping us. If they are sick, we are sick, and in many cases, they are helping us to digest the food and then to do all those useful stuff. And a lot of people, I know that we have a couple of folks coming from clinical pharmacology, and some of the bacteria, so they can help us to metabolize the drugs. And if they are being damaged, and then the drugs it will be not be as effective. So there are a lot of this type of research going on. So that's why that a couple of years ago, NIH put out this human microbiome project. It has been proven that so far it's almost related to every single disease that we have heard about. So it's super, super important. So here's the data access. This about human microbiome project. And again, the data is available. So they had two phases. The initial phase was to trying to see what are the microbiome species on the healthy individuals, very much like 1,000 genomes or GTAC. This offers you a reference. And the second phase now is more focusing on different disease. And they choose the pregnancy and the preterm birth and inflammatory bowel disease and type 2 diabetes as their current focus. Those are a lot of scientific discoveries out of this technology. Okay. We have another 18 minutes also. Okay. So I'll quickly go through this particular story in terms of how this technology will help a patient. And so this one is the story of a little kid. So let's go through this. So this is one of the very early phase in the genome sequencing technology. And there's a boy who presented at 15 months with this one disease I cannot pronounce. And it's progressing to something I cannot pronounce. And translate that into English, that will be severely underweight. When he ate, unusual holes would open between his intestine and skin, causing feces to leak into a large wound in his abdomen. And had more than 100 trips to the operation room in 2 years. The surgeon removed his colon and remained helpless. So the age and severity of this presentation suggesting there's some genetic mutation going on in this kid. So they want to figure out what is that. So this is the letter from the doctor to the medical school dean. This is from Milwaukee Medical College of Wisconsin. I'm writing to ask if there's some way we can get his genome sequence. There's a good chance Nick has a genetic defect. It's likely to be a new disease, and so on and so forth. But translate this into a scientific report. What they want to do is into a proposal is to identify the causative mutations actually through whole exome sequencing to provide necessary additional information for clinical management. That's what they are trying to do here. They eventually use the technology. Now will cost us about $400, and they did the whole exome sequencing on this kid. And because of this is an earlier case, the technology was not matured yet. They only identified 16,000 variants in the exome regions. So they have done a lot of very, very in-depth analysis. But the point is they were able to identify a hemizygous missense mutation in the X-linked inhibitor of apoptosis gene. And this is the cause of this particular disease. And eventually what they gave is they gave the cord blood transplantation to this kid. But you can see that after 7 weeks, and the boy is completely cured. So this is one of the cases that Francis Collins presented to the Congress to demonstrate how important scientific research is, and this how it helped the patients. I mean, I have to say this is an incredibly lucky case. Right, so they were able to identify the variants, and more important, there's a cure for that. But I think, if you think about deeper, this is a very generic treatment. It's not specific for this disease. But now we're getting much better. I'm pretty sure that everybody heard more than enough about the precision medicine, and this is being used in cancer a lot, and through the genomic technology, identify the variants and then select the individual treatment based on the variants. And that is something that's going on more dramatically now. So this is being everywhere now. So every major hospitals, including IU, have a precision medicine or personalized medicine clinic. And I'm pretty sure you guys heard about this Precision Health Initiative from IU, and it's trying to use established infrastructure to targeting so far it's 5 different diseases, and to look into the precision treatment and prevention space. Okay? Any question? It's a quiet group. I think some of the quiz is necessary to see whether you fully got it. Okay? So some of the very high level of concept, because I don't see that this is a concept that can fit in anywhere in the lecture, I decided to put here. So the first concept is super important. That is the barcoding or multiplexing. And the reason we need to do this is for most of the applications, we don't need so many reads per sample. In the past, when we run a sequencing instrument, we say, okay, we'll put one RNA sample into one lane, I will sequence it, and then I'll get the data. But now it's like each lane will give me 400 million reads. I only need 1/10 of that to profile the RNA sequencing. How do I solve this problem, right? And the way to do that is you will combine the 10 samples together to do the sequencing, but you have to, through certain way, know that which sequencing read is coming from which sample. So this is the barcoding. So most applications, we do not need to generate so many reads per sample, and we'll pull into a single lane of a flow cell, add a short index to tag the library. So you can think of, if I put 6 base in the beginning of every read from that sample, and for different samples, there are different 6 base pair reads, and then I can analyze those 6 base pairs eventually, know which read is coming from which sample. So that's the trick they are doing. And so for Illumina sequencing, normally it's a 6 base oligos, and then you can barcode up to 96 samples. And there is a more advanced approach that you can do dual barcode, which means that when we do the paired end, and one end we do the barcode, the other end we also do the barcode. That will offer you enough complexity to barcode over 300 samples into one. You can either push that by increasing the length of the barcode. So this is how it has been done. Actually, in the past, what we do is we use informatics to decipher which one it is. But now it's being integrated into the Illumina protocol. When they do the sequencing, they will automatically do it for you as long as you let the sequencer know what are the barcodes for each individual sample. So last time I gave a very much like a schematic view of how to do the analysis, but you can see here is the way they really do that is this is that tree, right? And it will become a forest. So you can see those are top and bottom. This is something called adapter sequence. And those are the 2 things that we put towards the end of the molecule, and that is needed. And then there's a couple of different colors. And this part, the middle part, is the DNA we want to sequence. Okay, and this is a few other sequence and colors. The yellow one, this is the sequencing, read 1 sequencing primer. So what that is you need that, and then the sequencing primer comes in when we do the synthesis, right? Sequence by synthesis. The primer needs to come in, and then the nucleotide will start to show up, right? So you have this read 1 sequencing primer. This is needed, and then the sequencer is going on. So after doing that, the next step, this is the barcode. This index is the barcode. Okay, this is 6 base pair sequences. So you can see we also have the reaction will do automatically. They will have another index sequencing primer come here, and then to read this. Oh, okay? So after doing that, if you want to do the paired end, okay, you turn the DNA to the other side. So you can see that this part is like the red is on top, and then you turn the DNA and then make it, the red is at the bottom, but completely rotate it, okay? And then what we do is we have the sequencing primer to target this and then to sequence the DNA molecule from this end. So this is something called a paired end, right? So we first got this DNA sequencing, we sequence this end, and then I somehow rotate it. There's a chemistry that you can do to do that, and then the sequencing primer come here and start to sequence this end. Okay, so now I got both two ends of DNA sequence. At the same time that the barcode or multiplexing index is being sequenced as well. Does that make sense? This is how they do that. Okay, so that's the first concept you need to keep in mind. There in this whole process, there's a multiplexing signal in that as well. The second concept I want to deliver is the paired end or mate pairs. So what's the difference of that? So first of all, single end sequencing. Okay, we got a DNA molecule. This molecule is like a 300 base pair each. The single end is by name you can know that we grab the DNA, we sequence from beginning, we only sequence one end of it. So the paired end is you sequence this end and then you sequence this end, right? Like what I showed in the previous slide, you make a turn and then sequence it. That makes sense? The mate pairs is different. Okay, so here is this would be good exam question about the differences between the pair and the mate pairs, right? So you can see here is the major differences. The first one is the fragment length is different. So for the paired end, this fragment length is similar to the single end. It's just you choose to sequence one side or two sides. But for the mate pair, this is a very long race, and normally DNA molecule normally is somewhere from 2,000 to 5,000 bases. It's very long range. And the second difference is you are not sequencing this direction, you are sequencing this direction, and from here to here. Okay, so that is the mate pair. So let me show you how this is being achieved. For the paired end sequencing, it becomes pretty straightforward. Okay, you got the DNA molecule, and then you got what the adapter is, right? And then I showed you have the sequencing primer, you sequence this end, you make a turn, and then you got another sequencing primer, you sequence the other end. That's the end of it. For the mate pair sequencing, this fragment is very long. So what you do is you will, among many other things, is you will do the biotin end on this two end, and then you make a circularization. Okay, you circularize it like this, and then you further fragment it, and then you use the biotin to capture your biotin. So by the end of the middle part, you are throwing them away. Okay, they are not being in. So like here, it may be somewhere around here, and it's going to be thrown away because there's no biotin. But for this part, you will get a molecule that is carry a part of here, carry a part of here, and they are being put head to head, okay, or tail to tail, depends on which part you like. And then we now have a much shorter fragments, and then you do the same way to put that on the adapters. So this is called mate pairs. Why we have to go through the trouble is if you do the sequencing identification, if you put the paired end plus mate pair together, it will help you dramatically to identify the structure variations, or you can do the de novo assembly much better. So you can see that this is a short read sequencing, and we sequence this end, and this will be different. But when you do the de novo assembly, normally what we do is, we normally don't do that anymore. But what can be done is, if you really care about the de novo assembly, is you use a mate pair protocol to sequence a small percentage. You have a low coverage of the mate pair protocol, and then you use the traditional protocol to sequence much deeper. So now you have the sequence accuracy being taken care of. At the same time, you have the larger structure variability in that. Okay, so that is what I need you to know about the mid-pair and pair end. The size of the DNA fragment is different. The directionality is different. And then I want you to know how mid-pair sequencing is being achieved. I think it's fair. Okay, so we're done. And for the next lecture, we'll focus on the data processing part. And I will briefly overview breaking the analysis into different components. And we'll talk about a Phred score, which is an evaluation for the accuracy of the sequencing, alignment, and the variant identification. And then I will focus, spend most of the time talking about sequence alignment in the next lecture. Okay? Okay, you guys have a nice weekend.